Hi, and a very warm welcome to everybody who's joining our webinar about enabling clinical-grade manufacturing of gene-engineered natural killer cells. I will talk today, as mentioned, about the manufacturing, but also discuss their analytics of current cases, the functionality of the current cases, and discuss further engineering strategies to overcome some of the limitations that current cases can still pose. And then we will come to the most important part of our webinar, which will be the discussion with our very valued panelists. So if we look at the adoptive cell and gene therapy pipeline, it is very obvious how most approaches that are out there and also who made it most far, as shown here in the middle for the marketed approaches, are actually those using autologous T cells, which are engineered either with a CAR construct or, for example, with TCRs. Natural killer cells have also been used in clinical trials already very often, and they have been used in the allogeneic setting as well and have shown that they are safe and can be applied in very high numbers even. And more now on the development pipeline upcoming are engineered NK cells, a lot of them engineered with a CAR construct for targeting. So why use CAR NK cells in cancer cell therapy? One of the reasons is the low risk. So CAR NK cells or NK cells in general have a low risk of causing graft versus host disease and can therefore be used in an allogeneic setting. They have a low risk of cytokine release syndrome, especially if we compare them to CAR T-cell approaches, where this risk is quite relevant. Furthermore, they have a limited risk of on-target of tumor effects because of their limited persistence. So for targets which are a little more risky, those could actually be the better choice. Furthermore, NK cells and CAR NK cells can be derived from an allogeneic source, so they can be used for patients who cannot undergo leukapheresis or have time pressure for their therapy, and they can be generated from multiple different sources. Furthermore, they hold the potential for off-the-shelf use. If we look at the anti-tumor activity, CAR NK cells actually have both features. They can have the CAR-guided anti-tumor action, but also have their innate immunity, so they can be active independent of antigen priming, and therefore they have the potential to prevent cancer cell antigen escape. One potential limitation is the duration of the response. NK cells and also CAR NK cells have a limited persistence after adoptive transfer, so they do depend on cytokine support for persistence. Currently, there is a list of clinical trials ongoing which use engineered NK cells in the cancer cell therapy. These include CAR NK cells derived from various sources, including the cell line NK92, including iPSC-derived CAR NK cells, cord blood-derived CAR NK cells, and peripheral blood-derived CAR NK cells. I'm now coming to our solutions for the manufacturing of gene-engineered NK cells. We are offering the complete workflow for your translational approaches. In our case, this would be starting with the leukapheresis material from a healthy donor, have a selection of highly pure NK cells based on magnetic labeling, activation of the NK cells, followed by transduction, expansion, and then formulation of the cell product for the sick patient. And these workflows include the medium, which is for the feeder cell-free natural killer cell expansion, and then the NK cell engineering protocol, which is GMP compatible, translational workflows, and then also the reagents in GMP standard quality. Our NK Max medium is available in research grade as well as Max GMP. And it enables natural killer cells to expand without having to use feeder cells in this culture. So the data shown here are from different donors. They have purified peripheral blood-derived natural killer cells as starting material. And this expansion here was done by adding cytokines and also AB0. Another cool feature of this medium is that the expansion of the natural killer cells is actually promoted as compared to the expansion of other cells, especially T cells, B cells, but also dendritic cells. And this you can actually see in these data here. So shown is the cell composition after 14 days of expansion. And the starting material for this expansion were actually PBMCs, so not purified NK cells. And you can see here that in the medium, the major fraction after this expansion is natural killer cells as compared to other media. And then, of course, very importantly, the effector functions are preserved after the expansion. I'm now coming to the genetic engineering of natural killer cells. Lentiviral vector-based gene engineering has been widely used for CAR T-cell therapies, and it has a lot of known advantages. Now, unfortunately for natural killer cells, this vector system showed low efficiency of transduction when used with the same pseudotype as for T-cells, which is the VSVG pseudotype. So if we take a closer look into this, it is not surprising. Shown here is the expression of the LDL receptor, one of the major receptors for the VSVG and protein. And as you can clearly see, T cells after activation highly express this on the surface, whereas NK cells don't. That is why we settled for the baboon envelope glycoprotein pseudotype lentiviral vector system. They use a different surface expression marker for recognizing the cells they transduce, and these are the ASCT1 and ASCT2 proteins. Shown here is a Western blot for the ASCT2 expression, and you can see that NK cells after activation highly express it. It is also expressed on activated T cells and on the NK92 cell line. The transduction of NK cells with the VSVG pseudotype lentiviral vector is shown here, is very, very low in its efficiency, if it's there at all, whereas with T cells, as known, it is very nice and efficient. And this is also shown here in the flow analysis data. Now changing to the baboon envelope glycoprotein pseudotype, you can see that with activated T cells and NK cells, there is a very high transduction efficiency. Here in these examples, it's around 70%. Please note that the activation of natural killer cells is required for that. We use IL-2 and IL-15, and also we add, for the beginning of the culture, an additional IL-1 family cytokine. Once we settled for the lentiviral vector pseudotype, we also aimed at having a protocol ready that gives an efficient transduction to be able to save lentiviral vector and use as low amounts as possible. And one way to achieve this is to do the transduction in the presence of vectorfusin. You can see here the difference. So shown here is the CAR expression on primary natural killer cells, and without vectorfusin under the vector MOI we've used there, it was around 20%, and then in the presence of vectorfusin, this was about 85%. Another way of increasing the efficiency of the transduction is to add a step of spinoculation. And this can nicely done in the Prodigy in our closed system because our cultivation chamber is also a centrifuge at the same time. Now, please keep in mind that the number of CAR expressing NK cells in your culture is not the most important point. It is also very important to take a close look at the vector copy number that you get in your settings. So the authorities require that you stay below a vector copy number of 5. And you can see here in black how if you increase the MOI of the lentiviral vector, the vector copy number logically increases, as does the transduction efficiency. And under these settings here, we would settle for an MOI of 1 or even 0.5 to be well below the vector copy number of 5, but yielding us a transduction efficiency going around 50%. The manufacturing of gene-engineered natural killer cells is a very complex procedure, and we aim at providing you with a complete and comprehensive solution package for both the manufacturing on the Prodigy as well as the corresponding analytics package for the Max Quang analysis, what we call in-process control and quality control, so IPC-QC package. We did develop the panels for the IPC-QC and also the express modes, which are automated gating strategies to enable the harmonization. All data that you have seen so far were from the research setting, so in the manual scale and small scale. And now I would like to walk you through the data that we received for the manufacturing in the Clinimax Prodigy. And I would like to walk you through all steps, so the selection, the transduction, the expansion, and then the cell product. The selection is done by actively depleting CD3 positive cells because for the allogeneic setting, it is important to have a very, very low number of T cells left. And then this is followed by an enrichment step with CD56 positive cells so that you have pure and viable NK cells. Following the activation of the natural killer cells in presence of the mentioned cytokines, we perform the transduction then on day 2 in the presence of vectorfusin and also including the spin inoculation step. The transduction rate is high and also stable, as you can see here on day 7 and on day 14. The average here is about 50%, and each dot here represents a single leukapheresis, so each one here is a different donor. Here's one example for the expansion of the current cases in the Prodigy. The triangle represents the total natural killer cell count, and then the circles here, the closed circles, represent the CAR NK cell count. And also shown here is the frequency, so the percent CAR positive cells within this culture. The cell product has a very high purity and viability, and also here the number of contaminating cells is below 0.5%, which is specifically important for the T cells and the B cells in the cell product. The natural killer cell and CAR NK cell count in the cell product is shown here. So the total NK cell numbers are in the average of about 2 times 10 to the 9, and the CAR positive NK cells within these average around 5 times 10 to the 8. And these are the numbers for the current process, which is in the standard sized chamber for the Prodigy. We are aiming to release this in Q1 next year. At the same time, we started the development of the next generation NK cell transduction large-scale process. So this is what the LS here stands for. There we aim at a current NK cell yield of 10 to the 10. As briefly mentioned before, the manufacturing of engineered cells is a very complex process, and a lot of different systems have been used and were also shown to be suitable. However, a lot of these require several manual steps, and this poses a risk of contamination, a risk of human error. And our strategy here is really to automate all these critical steps by having them in the closed system in the Clinimax Prodigy. Now, if you are looking into off-the-shelf and large-scale NK cell expansion, how can you make use of this automated closed system? So there are two options. One option is that you do all these separation and gene engineering steps within the Prodigy, then connect this to your bioreactor system. And you could even also connect this back to the Prodigy to have an automated formulation of the cell product. And for this, actually, if you're interested in that, please contact our customized application process team. You can find the information at the website. Another option would be that you generate the current K cells in the Prodigy and then you freeze a master cell bank, which you could use as seeding material for your bioreactor. I will now talk about our analytic solution for the current K cells. As many of you are probably aware, setting up the analytics for a cell manufacturing process is actually as much effort as setting up the manufacturing itself. So it's important to really not underestimate this part. To facilitate the harmonization of the analytics in the cell and gene therapy, especially between different centers, we implemented the express modes into the Max Quant. Express modes are automated gating strategies, and they were designed as a tool for the standardization of your data analysis. The advantages are that they are easy of use, time-saving, reduce the human error, and as mentioned, they are for standardization and reproducibility, but also enable an easy documentation. Here is an overview of our analytic strategy. For the donor selection, we have antibody panels to look at the KIR expression profile of the natural killer cells, also available with the corresponding express modes. And then for the in-process control and quality control for the manufacturing of the CAR-NK cells, we have the IPCs on day 0 on the starting material, as well as after the selection of the natural killer cells. And then there's one in-process control on day 7 to look at the transduction status of the cells already. And then the quality control will be done on the cell product. So the antibody panels and the express modes for the cell composition and the cell transduction will be available also in Q1 next year. Starting from this in-process control, the CAR detection reagent becomes important because it is about looking already at the transduction efficiency during the process and in the cell product, and then also later during patient monitoring. This, of course, will be of importance. Our IPC and QC panels are, on the one hand, for the cellular composition, which looks at the viability and the purity of the natural killer cells. And the release criteria here would be the amount of CD3 positive and CD19 positive cells, in addition to the viability. And then the transduction efficiency panel, on top of that, has included the CAR detection reagent. So this is a reagent that specifically detects the CAR on the surface after transduction. And here the release criteria would be the percent CAR positive within the cell product. So as mentioned, these will be available early next year. And then we are working also on panels for the concomitant research activities. Those are panels looking at the maturation, activation, as well as inhibition status of the natural killer cells. In case you are curious to find out more, please come to our website where you can find the detailed protocol for the IPC and QC of the CAR-NK cells. And here you can also take a look at the exact composition of those panels. I would like to give you a very quick summary about the CAR detection reagents. So we have two types. Either they have been raised as a monoclonal antibody against the CAR, so the anti-idiotype antibodies, or they are a extracellular domain fusion protein. Both CAR detection reagent types have a mutated IgG1 Fc, human, of course. It has been mutated so that it will not bind to Fc gamma receptors and you will not need any Fc blocking. And here you can see an overview of the ones that are available right now. We have more in the pipeline as well. And these, of course, we have also tested for the natural killer cells. Here you can see an example for the CD19 CAR detection reagent, the untransduced natural killer cells in comparison to the transduced natural killer cells with the CD19 CAR. Now I'm coming to the next agenda point, which is the functionality of the CAR-NK cells. The functionality data that I am going to show you now were all obtained from CAR-NK cells that we have manufactured in the Prodigy, as I have described to you before. And we used a specific cell line as a test system, which is an human acute leukemia cell line that is resistant to NK cell innate immunity. Because NK cells, as they are natural killers, they can kill most tumor cell lines out there. So we screened for a cell line that is mostly resistant because it has a upregulation of ligands against inhibitory receptors and downregulation of ligands for certain activating receptors. And you can see here the cytotoxicity of the natural killer cells and also the CAR-NK cells in co-culture with this tumor cell line. And shown here is always the person's cytotoxicity. And so the tumor cell line here is resistant. Not for every donor, it is as resistant. Some NK cells actually are a little more active, but we can always see a very good delta between the non-transduced and the CAR-transduced NK cells. So if we transduce the cell line with the tumor target, the untransduced NK cells cannot kill it, and then the CAR NK cells very effectively and specifically kill those cells after the tumor target has been introduced into them. And this shows us two things. On the one hand, that the CAR recognition is very specific, and on the other hand, that the CAR can actually overcome these inhibitory effects here towards the NK cells. The same is mirrored in the cytokine secretion of the CAR NK cells. So what you can see here is the same setup actually with the effector cells and the target cells in a ratio of 1:1. And for the cytokines GM-CSF, interferon alpha, and TNF alpha, you can see that the cytokine release is actually increased. If we look at a bigger array of cytokines, you can see that natural killer cells, as they are also known, are safe. So it's mostly those three here that are being secreted. And interestingly, CAR T cells actually secrete higher levels of cytokines in comparison to CAR NK cells. And this is shown here at the example of GM-CSF. Here the CAR T cells and here the CAR NK cells. We also tested the CAR NK cells after manufacturing in the Prodigy in an in vivo model with an ALL cell line in the mouse. And we did inject IO2 on day -1 and then over a time span of 7 days to look at the persistence of the CAR NK cells. And this is something that you can see here very clearly. So in green are the natural killer cells without the CAR. They show some little tumor control here. And then the CAR NK cells have a very efficient anti-tumor activity as long as the cytokine is provided. And then after stopping the cytokine supply, the persistence of the CAR NK cells is limited, and you can see how the tumor here relapses. And this here is just an image from day 8 of the mouse experiment. This brings me to our next topics, the challenges of CAR NK cell therapy. So despite manufacturing and functionality, one of the major challenges is the persistence after adoptive transfer. Furthermore, the activity of the CAR NK cells and also the situation that you can have if you use a tumor target that is also expressed on the NK cells or upregulated during the culture. Then you have a phenomenon which we call fratricide, when the CAR NK cells actually kill other CAR NK cells or NK cells. And there are different ways to how this can be addressed. One very important way, of course, that we have Rizwan here in the panel, I would like to mention, is the simulation with a certain cytokine cocktail. Other ways would be to co-express the cytokine within the CAR construct. And what I would like to talk more about now is the knockout strategies or knock-in strategies of immune checkpoints. To be able to further engineer the CAR NK cells, we developed a protocol for the electroporation of the natural killer cells. As shown in this example from our CAR T-cell team, both the transduction as well as the electroporation can be done within one Prodigy run. Here you can see an example from the CD19 CAR T cells. So the T cells are selected and then transduced with the CD19 CAR and then electroporated with TALEN mRNAs for the T cell alpha beta locus. If you're interested to see more data about this, I provided here the reference, and I could also send you the paper if you let me know afterwards. To develop the protocol for the electroporation of natural killer cells, we made use of the high flexibility of the Miltenyi electroporator, because you can really play around with the settings and, very importantly, with the pulses. This enabled us to find settings where we can have a high electroporation efficiency, as shown here. We had more than 80% GFP positive NK cells, yet still retaining a high viability. And here on the right-hand side, it is shown that the GFP expression after electroporation was quite stable over time. Looking into the expansion capacity and effector function of the natural killer cells after electroporation, we were actually happy to find that the NK cells are not impaired as a consequence to the electroporation. So the expansion capacity is still retained, as well as the cytokine release after co-culture with tumor cells and the cytotoxic activity. With this electroporation protocol, we have meanwhile performed the CRISPR-Cas knockout of a few different markers. And shown here is an example of one marker that is very highly expressed on the primary natural killer cells, here up to 90%. So MOC-KO means that the electroporation was performed, including all ingredients except the RNPs. And then KO is the knockout sample where the RNPs were included in the electroporation sample. And it is clearly shown how we have a very efficient reduction of the surface marker. And here the same results are shown in the bar chart in triplicates. With this, I would like to thank you very much for your attention. We are now coming to the part that I am most looking forward, which is the panel discussion. I am hoping for a lively discussion and look forward to your questions. Thank you very much.
Thank you, Nina. Great presentation. So now let's move on to the panel discussion. At this point, we're going to welcome Rizwan and Evelyn. So I hope you're both ready to answer some questions.
Yep.
Brilliant.
Yes. Hello.
So, brilliant. So we'll get started then with what are the main advantages of using NK cells in cell therapy? Rizwan, do you want to start us off on this one?
Yeah, no, this is my favorite question because I get to stress the, my love affair with NK cells. So, I think the biggest advantage, as Nina mentioned briefly in her talk, is, we don't see cytokine release syndrome, which, is a big problem still, despite all the advances in CAR T cell world. And then more importantly, neurotoxicity as well. In my opinion, those are the major two advantages of using NK cells. And then furthermore, I think what I really like about NK cells is that they maintain the both CD16 as well as other non-CAR mediated killing of the tumor cells, which you can still harness by combining, for example, anti-MICA, MICB antibodies, so you can engage the CD16, and some of the other checkpoints, some of the other activating receptors can still be engaged, say through bikes, where you are engaging those active like NKP46 and things like that. So it's like a dual targeting the CAR, but also you have the endogenous receptors which you can harness. So those are my three main reasons why I think NK cells have advantages, and I personally think that NK cells are better than T cells. Evelyn?
I completely agree with you, which is no question. I mean, one issue that I like very much when working with NK cells is also their intrinsic anti-tumor ability. So even if you think on engineering T cells for adaptive anti-tumor therapy, NK cells even function in absence of antigen expression and have an intrinsic anti-tumor effect based on this balance of activating and inhibitory receptors, especially in protocols where NK cells are activated with cytokines. And in meantime, they have then their low risk of GvHD, and that's really amazing to think about that we can engineer our NK cells, that we have a CAR-mediated anti-tumor effect that's very target-specific, but in case of antigen loss, also we would profit from the intrinsic anti-tumor effect.
And Evelyn, may I add something? You mentioned the lack of GVHD. One of the challenges in immunotherapy in general has been this on-target, off-target, off-tumor, toxicity. And NK cells maintain and that applies to NK cell cause. Although they have the gears, they have the other means of smelling, if you will, abnormal from malignantly transformed cells. So potentially, not only they don't have GVHD because they don't cause necessarily alloreactivity against normal healthy tissues, but then even in say autologous fashion or third-party setting, not just GVHD, but also they can, even if they are armed with a CAR, say, for example, mesothelin or HER2, where HER2 or mesothelin may be expressed by normal healthy tissues, at least theoretically, again, there's some preliminary data, but not a lot, but at least theoretically, there's possibility of NK cells maintaining those checkpoints where they are still able to not be activated as much as by the malignantly transformed cells.
Yeah, exactly. That's a well taken point, and enables maybe to use NK cells then for tumor entities that are difficult to target, such as acute myeloid leukemia, where you don't have such a very specific tumor target, and the surface molecules are also expressed on healthy hematopoietic cells.
Absolutely, that's a great point. And Nina, what are the new trends to engineer NK cells?
Thanks a lot. I would actually also like to add to the last point, but it's actually, it goes for both. So I would like to add the point of view of manufacturing, what in the main advantages of using NK cells, because we all know that despite the remarkable progress that we have seen with CAR T-cell therapy in cancer, now with already FDA-approved products currently on the market, its autologous nature still limits the patient's accessibility. So in an attempt to address these limitations, more and more the field is turning towards allogeneic platforms. And T-cells, of course, are not ideal sources for the allogeneic therapies due to their HLA-dependent recognition and their potential for inducing GVHD. And this is another factor why NK cells are really a safe and suitable allogeneic source for also off-the-shelf cellular immunotherapy aspects.
Definitely. And Rizwan, do you have anything to add on what are the new trends to engineer NK cells?
I think the advances in iPSC technology, although you could theoretically apply iPSC technology to both NK cells and T cells, and there have been there's some preliminary data that you can generate T cells, but so far it's mostly NK cells which have been generated from iPSCs, and that also gives a huge advantage to the NK cell field because you can do multiplex, multi-edits, all sorts of engineering in the iPSC cells, and then you can derive unlimited numbers of NK cells. That also makes NK cell field very attractive.
Absolutely. Anything to add, Evelyn?
I mean, concerning engineering of natural killer cells, I think we have improved technologies a lot in the last years to be able now to engineer primary NK cells from different sources as well as iPSCs. And I think Nina has already presented the possibilities of both, that we have viral transduction and also non-viral procedures for an efficient, stable transduction of NK cells. That helps a lot. And in the beginning, it was very difficult to overcome limitations to transduce natural killer cells because the VSVG pseudotyped lentiviral transduction protocols worked really much better in T cells. But then in the last years, we learned that NK cells express just other entry receptors, and they are quite suitable when the envelope is different. So pseudotyping with RE114, as we found in our group, or the Boone pseudotype lentivirus works very well in NK cells.
Thank you. And what are the challenges of efficient transduction of NK cells? I think we already touched on that a bit there, but Rizwan, do you have anything to?
No, I agree with Alan. This is exciting. These are really exciting times in the field because finally that doesn't seem to be a major challenge anymore, especially with the baboon and some other, unique glycoproteins which we use to serotype the lentivirus system, and also retrovirus. For example, RD114 also goes through the same ASCT2 receptor, so you can use RD114 and get the retrovirus in. And there's some data that you can use actually RD114 on lentivirus, and actually that works as well in some studies. So you have a bunch of options. I think it was kind of naive from our side, several years back when we just simply used the VSVG, thinking that it should work, when in reality, NK cells are not the same as T cells, and as expected, we had to tweak and custom made the envelope glycoproteins with serotype these lentiviruses. So I think we are finally at a point where really transduction is not a barrier anymore, and which is awesome.
That is awesome. And Nina, do you have anything to add on challenges of efficient transduction of NK cells?
Yes, of course. So, we have focused our efforts towards the lentiviral vector-based transduction because of the named advantages, right? That the clinical safety has been proven, the transduction is efficient, and even it's able to transduce non-dividing cells. And as shown, and also as Rizwan has mentioned, now changing the pseudotype really helps transducing natural killer cells. Now, and this goes for every new pseudotype, new challenges come then by changing the pseudotype, because if you have a lentiviral vector production system that has been optimized for one pseudotype, things can change. Yeah, the production protocol may have to be re-optimized. Maybe the titer determination is not working as efficiently as for the established pseudotypes. So there come new challenges with that, of course. So the production protocols and the QC protocols for the lentiviral vector itself will have to be optimized again. And then once you have defined a lentiviral vector system, it is still very important to invest the effort on defining the exact specifications for each new construct that you are introducing. So meanwhile, we don't have to change the general manufacturing protocol anymore, like adding vector-fusin, including a step of spinoculation, we still have to take a close look into new constructs with regards to the producibility, which MOI to use, the transduction efficiency, the vector copy number, the stability on the surface expression, and then also in vitro and in vivo functionality, because if single chain variable fragments for the same target are introduced in the same CAR backbone, but the single chains themselves differ, this can have a complete different effect in vitro and in vivo. So also there must be a lot of screening for the best CAR construct, as is the same for antibody therapy, of course. And then not to forget, you always need a CAR detection reagent, which has to be produced, and it is very important that it's specific and really able to stain the CAR expression on the surface of the NK cell. And depending on the nature of the target, it can be easy to pre-produce, but this can also be very challenging in itself.
Thank you. And request here for some practical advice. How do I translate my research workflow into a GMP grade manufacturing process? What are the key considerations, best practices in transitioning from an open manual to closed automated bioprocessing? So what are the challenges and gaps there? Nina, do you want to kick off with the Miltenyi perspective on that?
Yes, thank you. So to translate the research workflow into a GMP-grade manufacturing process, our vision and strategy is to shift away from an open manual process towards the closed and automated system, so to reduce the open steps as far as possible, because each manual step poses a risk for contamination as well as for human error. Each manual step is time intense in the clean room.
And then one more strategy would be to reduce the required clean room class as far as possible to reduce the cost and efforts to maintain the clean room. So in the US, for CAR T-cell manufacturing in the Prodigy, the recommended clean room class is C. And in Germany, we are currently working towards approval for clean room class D for the Prodigy, for the engineering in the Prodigy. Then, of course, the manufacturing protocol must be highly reproducible under the situation of donor-to-donor variations. So it has to be set up in a way that it is ensured that efficient transduction and expansion is obtained from every donor. Another important point is that all ingredients have to be GMP compatible. So to already set up the research use only processes with reagents that are available as GMP. Yeah, and then I believe another very important challenge is the timing of the manufacturing. So to reduce the steps which require actually people in the clean room as much as possible, because everything takes like at least 3 times as long in the clean room as compared to the normal lab.
Absolutely. Thank you, Nina. And Rizwan, do you want to talk a bit about how you've gone about this process?
Yeah, no, Nina, I agree completely with Nina. When we start, so I moved a few years back from WashU to Dana-Farber to lead the NK cell program here. And I thought it would be super easy. We have an outstanding GMP facility, but then I realized that, starting something from scratch is challenging. And, even though we had a fabulous GMP facility, unless you have experience with NK cells, there are some unique tweaks you have to make sure you do before everything works, and so the validation runs and things like that. But in terms of CAR NK cells, I think the biggest challenge in terms of manufacturing, for translating from preclinical work to clinic, is actually finding a vendor which can make a GMP-grade lentivirus. And then if you are, you want to use baboon-based system, then it is even more challenging, because most of the vendors which make GMP-grade virus don't have much experience with it. As a matter of fact, as far as I know, Miltenyi is the only vendor which has experience with baboon-based lentivirus system. So that's been one of the biggest challenges for us personally. We have a product almost ready to go to clinic, but it's a long waiting time, and getting access to the GMP grade lentivirus system. And then obviously once you have that, then you can do some validation runs and then apply for IND. So it's a long, lengthy, and expensive process. I think Prodigy system is really, really attractive. As Nina mentioned, that if we can downgrade the clean level, that will make everybody's life easy. And otherwise, it takes incredibly long time to manufacture these cells, and is very labor intensive currently. So I personally think that Prodigy and any other similar system which uses closed manufacturing, these are the kind of things which will be really helpful for both CAR NK cells and T cell fields.
Thank you, Rizwan. Evelyn, any advice from Frankfurt?
Mm-hmm. Yeah, these points are well taken. For sure, it's of high importance to get an excellent GMP grade virus production to have the capability then to produce a large amount of CAR NK cells. Or this will take us to this other aspect that we mentioned in the beginning and has been shown in the webinar by Nina. That new technologies also emphasize on non-viral systems that could also be used in the Prodigy closed system combined with electroporation protocols. And I believe that besides the RNA-based electroporation of NK cells, also other technologies, for instance, to introduce with transposons, which are jumping genetic elements, the genetic information via technology called Sleeping Beauty, that's also well known for CAR T-cell production, and in first clinical trials that could be also used then for non-viral transduction of NK cells in such automated systems. And I think what's an important aspect, this could be less costly and also has been reported to show very safe genomic integration profile, which could be an advantage. But what we will need is to really find markers and profiles for the quality check of the resulting NK cell products, independent if they are viral or non-viral transduced and engineered cells. It will be of high importance at the end of the procedure to have a QC based on flow cytometry or mass spectrometry or PCR, and hopefully to find predictive markers to know the impact on and the functional quality of the resulting cell product.
Thank you, Evelyn. And regarding the analytical tools, what is the current state of the art and where do you hope to see further innovation in this regard? Rizwan, do you want to kick us off there?
Yeah, no, in terms of, the current kind of work outflow we have for analyzing our products before they're released are, manual flow, and that's, again, very cumbersome. And, every time a product is released, we take some cells at different time points, actually right before they infuse the patient, but also prior, as Nina showed that, she showed like at every point, basically you can take some cells and analyze them. But that again is cumbersome and it delays things. And we use the markers she mentioned, we use the viability, we use things like that. But it's very manual, and that brings a lot of, again, that every step you add a manual step basically means delay and labor intensive. So I think in terms of how to make life easier for everyone, if you can build in these analytic tools where basically you have the capacity in the closed system that it can take a sample and internally and stain for, say, key elements, including, say, transduction efficiency, viability, and things like that, and generate a number and which meets the threshold, that will make life easy for everybody. So that's in terms of QC analytical tool. But then on the other end, which is after you adopted transfer NK cell cause into people, what methods to use to track them. And again, Nina mentioned that, they have developed the antibodies against the idiotype where you can track them. But, you can also put certain tags on the car where you can use antibodies that against those things to monitor the what happens to the NK cell. I think it's very critical that we shouldn't forget about investing into developing, detailed tools or detailed analytic tools which will allow us to track these cells and what happens, how long they last, what happens in terms of exhaustion, what happens to, are they able to track in the tumor site. So good correlative science is very, very important, and we should, because again, NK cells are probably 10 years behind T cells, and there's a lot to learn. I'm not saying we have learned everything about T cells, but I think NK cells, there's a lot of unknowns right now, and having the right analytical tools to allow us to do deep dive after their adaptive transfer is also, in my opinion, very, very important.
Absolutely, exciting times. Evelyn, do you have anything to add there?
I just want to highlight again, as has been mentioned, that in the very end, in the future steps now, I hope we will have the chance to further learn to engineer NK cell products to improve functionality and also migration and persistence of these NK cells.
Nina, I'll give you the last word there.
Yes, thank you. So, I think so by developing the CAR T cell therapies, which is all, which Miltenyi has also done, but also a lot of other companies and academias. And I think what we, and I think also many others have learned, that establishing the analytics of a gene engineered cell product is at least as much effort as establishing the manufacturing process itself. And this is something that I think a lot of people, including ourselves, have been guilty of underestimating. So to help with this, it's really important to have a sort of automation also in the accompanying in process control and quality control of the cell product. So to have automated gating strategy to enable harmonization between centers, but also to have defined panels for each cell product. And the next step would be to have premixed antibody cocktails or even freeze-dried formulations of the same, so that this would be even more standardized. And then one important aspect of NK cells versus T cells, of course, is that in the quality control, we have the rare cell analysis for the CD3, right? Because this is, in the allogeneic setting, one of the risk factors. And this is also something that can be more time consuming than in the end for the final QC of the cell product, because a rare cell analysis requires that you have to count quite a volume to be sure that enough cells have been counted to give a really reliable answer on how many T cells are really left there. And also there, it's very important to have a panel that is very reliable here, does not give any background, because this T cell count is really, it's key in the allogeneic setting to have this reliable and no false positives and no false negatives.
Absolutely. And a viewer here asks, says that they're curious to hear the panel's thoughts on the advantages of using NK cells from cord blood versus peripheral blood for cell therapies. Nina, do you want to start us off there?
Yeah, sorry, I was still muted. So I think, many sources for allogeneic NK cells are available, right? Including NK cell 92 cell line, PBMC derived, hematopoietic stem cell derived, induced pluripotent stem cell derived, umbilical cord blood derived. And I believe that each source has its advantages and limitations, but overall they do share one benefit, which is that they can all be produced for allogeneic cell therapy, including the reduced manufacturing time, the increased feasibility for large-scale production, and everything else that comes with it. When it comes to directly comparison, I have to say I would be more interested in what Rizwan or Evelyn would have to say, because our approaches have so far focused on peripheral blood-derived NK cells. We are working on other sources as well, but our main experience is there really with the peripheral blood NK cells. So maybe Rizwan or Evelyn, do you have to add? Can you add more to the direct comparison?
No, first of all, I agree. It's always good to have more options than not, and it's a good problem to have that we have cord blood, we have peripheral blood, we have some cell lines, we have iPSC derived. So all of these are great, and also some placental derived. So this is fascinating that seeing multiple options. Now, more specifically about cord blood, I personally like cord blood because you have untapped hundreds and thousands of cord blood units already stored and HLA typed. So if you can tap into that resource, I think that's fabulous. Personally, though, I'm biased because my group primarily uses what we call memory-like NK cells, which are generated from peripheral blood, and that's what we have the experience most with. So, we have shown that these memory-like NK cells, which are easy to generate and differentiate from the peripheral blood, they can persist for months in an immune-compatible environment and also expand, which in my opinion is a great advantage. And obviously, we are now arming them with CARs. But having said that, it doesn't mean that, cord blood derived NK cells or NK cell CARs are not good. I think, as I mentioned, we have hundreds and thousands of banked cord blood units, and if someone were to take advantage of that, I think that's great. But I don't see any direct head-to-head comparison study that I know of. In our hands, we did try to generate memory-like NK cells from cord blood, and we could not see as much robust activity of memory-like NK cell derived from cord blood as from the peripheral blood. But that's in our hands.
Thanks, Rizwan. Evelyn, anything to add there?
I would like to add that we should really have the focus on the benefit for the patients, and I believe that from the CAR T cells feel that CD19 CAR products work really great, but it happens that the generation of autologous CAR products fails due to the fact that patients are heavily pre-treated and the immune cells don't expand as expected and as needed. And in this case, different sources can be used then to have off-the-shelf NK cell products that are firstly produced and from third-party donors, for instance, also after allogeneic stem cell transplantation or in haploidentical settings. And this is really a great advantage for the patients.
Brilliant. Okay, we're coming up towards the end of the webinar, but we try and squeeze in one or two more very quick questions. So for Nina, why do you transduce the NK cells prior to expansion?
All right, thanks a lot. So the reason is that to save money by using as least amount of lentiviral vector as possible, we try to transfuse as early in the culture as possible.
Makes sense. And another one, I think, this one's for you as well, Nina. Why is ASCT2 expression shown with WB but not by FC?
So why is the ASCT2 expression shown with Western blot? Why not? I believe it's meant for flow cytometry, right?
Yeah.
So at that time when we did the experiments, we could not really find a good antibody against that target, which was also suitable for flow cytometry. So I very, very much prefer, of course, flow cytometry over Western blot, and meanwhile we found a better one. If the person who asked the question is interested, feel free to email us if you're interested in the antibody. I can share which one we acquire there.
Brilliant. Well, thank you all for answering the questions there from the audience. That is all we've got time for today. But any questions that we didn't get to today, we will try to reply to those by email. So if we didn't get to your question today, don't worry, you should still get an answer. The webinar will be available on demand tomorrow, so look out for an email from us with the link. And I think all that's left then is to thank Nina, Rizwan and Evelyn once more for a great webinar. And thank you to the audience for listening. We hope you'll join us again soon. Goodbye.
Thank you. Bye.
Thanks a lot. Bye-bye.